very blue here with the phone cameras today. Welcome to Dr. C's Excellent Science Adventures. We're in a secret location near Orlando, Florida today, looking at a spectacular facility that's breeding out stony corals. Marine animal rescue usually brings to mind tending to injured manatees or sea turtles. So why exactly should stony corals need rescuing? Stony coral tissue loss disease is a mysterious wasting disease that appeared in stony corals off the coast of Miami in 2014. This animation from NOAA illustrates the rapid spread of the disease throughout Florida's coral reef systems. Beginning in September 2018, the Florida Coral Rescue Team started collecting unaffected corals to preserve genetic diversity and serve as future breeding stock for the 20 species of coral affected by the disease. The Orlando Center is one of 18 land-based facilities in 12 states established to maintain and propagate the rescued corals. Evan and Lexi, two of the center's staff members, generously took time to explain the workings of the facility during our visit.
Corals are in the same day-light cycle they would be in nature. We also have moonlight simulators, which are the little LEDs. They're off right now. But whenever there's a...
Oh, that's cool. Yeah.
It's full, they're on all night long,
okay.
So the corals can synchronize their spawning with the wild. We come in here to the facility at the same time they're supposed to spawn in the wild, and we'll watch them spawn. We'll turn the flow off kind of like it is now. It'll usually be dark. A couple corals spawn before darkness, but most of the corals spawn at night. We'll come in, watch the corals, the eggs bubble up, collect the eggs, fertilize them, and then we'll either send them to other facilities. We're looking to get more room right now.
Corals are very sensitive to water chemistry and microbiome. Each of the tanks includes several chunks of living rock, a piece of limestone that was left in the ocean for a couple of years in the Florida Keys to be colonized by the natural biota.
So we test about, I would say on average, about 10 different parameters every week. We test temperature, salinity, pH, alkalinity, calcium, magnesium, phosphate, nitrate, and then we do an ICP every month. We test every element that we can test for in the water, and so far we've had very good luck. We haven't had any problems with the water.
So the little anemones that are growing on this, those were in the ocean when
those
it was
were in the
pulled
ocean,
back out.
So the little anemones, the little sponges, everything that's on
the
there.
live rock is to seed
it comes in and it seeds
the ecosystem here.
The purple you see on the wall is actually an algae called coral algae. You can see the urchins will eat it off, and the purple color you see on the tiles and things is a type of algae called coral algae. But it's very simple filtration. The lights, so they come on at different times of the day, basically corresponding to Key West. Most of coral spawning takes place in late summer or fall, September, August, September time. So we'll come in, we'll collect a lot of the eggs and sperm. The pH inside here is 6.5. So we lower the pH in here, it dissolves the limestone. Basically, it's dissolving coral skeleton to replace the coral skeleton in the exhibit, or in the aquarium. So water slowly goes through this. We add CO2, which is CO2. The CO2 is bubbling right here. The CO2 is injected into the pump, and it brings the pH down now. It's amazing how fast just a little bit of CO2 will drop the pH in this. Throughout this, it'll dissolve the limestone, and then we drip that back into the tank. That basically will provide them enough calcium and alkalinity for their skeleton so they can continue to grow.
In the ocean, coral larvae drift with the currents until they fortuitously come upon suitable substrate on which to attach and begin growth. In the Coral Rescue Center, the odds of success are much higher. The larvae are released into a tank with small tiles perfectly suited for their attachment. This is time-lapse video Lexi took of coral larvae in a petri dish. It's amazing to think that each one of these things has the potential to grow into a coral head the size of a dinner plate or a bowling ball. This breeding program has been so successful that they're pressed for space in this facility. SeaWorld Orlando stepped up by renovating an exhibit hall within the park to accommodate additional grow-out tanks. Should you visit the park, be sure to have a look at this facility. Yeah, I know the orcas and dolphins are the stars of the show, but stony corals are key players in building the physical structure of marine ecosystems. By one estimate, Florida's coral reefs prevent $400 million a year in wave damage to coastal facilities, not to mention their value for biodiversity, fishing, and tourism. Other institutions are trying different approaches to reef preservation. Scientists at the University of Miami developed an antibiotic paste that stops the spread of stony coral tissue loss disease. This is much more labor-intensive than breeding and transplantation, but might preserve wild coral populations in selected areas. Branching corals are immune to the tissue loss disease and can be grown in large numbers, as is being done by the Coral Restoration Foundation. Unfortunately, the branching corals are more heat-sensitive than the other stony corals and are more susceptible to bleaching. Degraded coral reefs typically become overgrown with algae. Jason Spadaro at Mote Marine Lab has conducted field experiments demonstrating that Caribbean king crabs, voracious algae eaters, facilitate settling of coral larvae by clearing patches of the reef surface. A captive breeding program at Mote has a target of producing 250,000 juvenile crabs per year for release into the wild. Other scientists are focused on understanding heat tolerance in zooxanthellae, the symbiotic algae that give corals their color. It's encouraging to see so many bright, capable people working on reef restoration. This will be an interesting topic to revisit 5 years from now.